Let's talk about something that sounds like it's straight out of science fiction, building a human chromosome. Completely from scratch. I'm not talking about editing a gene here or there. I mean building the entire complex operating system of our cells from the ground up. It's a wild idea, but what was once just a thought experiment is now actually happening. Yeah, just let that sink in for a second. Back in 2000, this was a pretty shocking question for a leading scientist to ask in a major journal. It just goes to show you how massive and high stakes this whole field is. We're literally talking about creating the fundamental building blocks of our own biology. So here's our roadmap for today. First, we're going to tackle the big why, the grand problem that this technology could solve. Then, we'll look at the essential parts you need. We'll cover the incredible first breakthrough, then a huge roadblock they hit, and finally, the absolutely brilliant solution that's paving the way for the future. All right, section 1. Think of this as the quest for a new genetic operating system. This is all about why we'd even bother. This isn't just some cool lab experiment. It's about solving some of the biggest challenges in medicine. Which brings us to the main question, right? Why would we want to do this? I mean, beyond just the because we can factor, what's the real-world point of building a chromosome from scratch? Well, the potential is just staggering. The simple answer? An artificial chromosome could be the ultimate gene therapy machine. So think of your natural chromosomes as the cell's operating system, right? The software it runs on. A lot of current gene therapies use viruses to deliver genetic fixes, which is kind of like using a small, sometimes glitchy software patch. But a human artificial chromosome, or a hack, is different. It's like installing a whole new, perfectly clean operating system. You could load it up with massive genes, or even entire suites of genes, to fix really complex disorders like Lesch-Nahan disease. And because it behaves just like a natural chromosome, it sticks around, getting copied and passed on every time a cell divides, all without the risks that can come with viral methods. Okay, so the why is huge. But to build something, you need to know what parts go into it. And this is where scientists ran into a much deeper problem, a real fundamental puzzle they had to solve first. So when you boil it down, a working chromosome needs just 3 essential parts. You've got telomeres. These are like the little plastic caps on the ends of your shoelaces. They stop the DNA from unraveling. Simple enough. Then you need origins of replication. These are basically the start copy signals along the DNA. Scientists had a pretty good grip on those too. But that third piece, the centromere, well, that was the real mystery. This thing is the absolute engine of the chromosome. It's the control hub that the cell machinery grabs onto to pull the copied chromosomes apart during cell division. If you don't have a working centromere, your chromosome is just lost to the void. It's totally essential, and for the longest time, nobody really knew how it worked. It was a complete black box. So what do you do when you need an engine, but you don't have the blueprints? Well, the first genius idea was to just borrow one. Take an engine out of a car that you already know works. And that's exactly what they did. And in 1998, it happened. A team in Japan led by Masashi Ikeno published this incredible paper showing they'd done what a lot of people thought was impossible. They had built the very first functional human artificial chromosome. Their method was this super clever feat of biological engineering. First, they managed to snip out a big chunk of DNA, about 100,000 letters long, right from the centromere of a human chromosome. This was their borrowed engine. They then stitched that piece into a carrier molecule, slapped on some telomeres, those shoelace tips, and then just delivered the whole thing as naked DNA into human cells growing in a dish. And would you look at that? That tiny glowing dot right there inside a human cell nucleus, that's it. That's the first man-made human chromosome, and unbelievably, it worked. It copied itself and was passed down to new cells with over 98% accuracy. This was a massive, massive leap forward. But here's the twist. The very thing that made it work, that borrowed piece of a natural centromere, turned out to be its biggest weakness. It solved one problem but created a brand new one. It turned their amazing invention into a scientific black box. See, the problem is a natural centromere isn't made of some nice, clean code. It's a total mess of what's called alpha satellite DNA. We're talking about the same short sequence repeated over and over and over again for millions of base pairs. Imagine trying to read a book where it's just the word "the" for 1,000 pages. It's basically unreadable. You can't analyze it, and you definitely can't engineer it. So, the analogy is this. That 1998 breakthrough was like building a working car using random parts from a junkyard. I mean, the fact that it runs at all is incredible. But you have no blueprints, you can't tinker with the engine because you don't know how it's built, you can't use any of your modern diagnostic tools on it. To really get somewhere, you've got to move beyond the junkyard parts and start working from a clean, precise blueprint. And for nearly 2 decades, that black box problem was a major roadblock. But then, a new generation of scientists came up with a totally different and, frankly, revolutionary approach. Instead of borrowing an engine, they figured out how to build one by design. So this was the new puzzle. If the original blueprints for the engine are a chaotic, unreadable mess, how in the world do you build a new one? The answer is just so elegant. Instead of trying to copy the messy old engine, you just hijack the cell's own factory and tell it exactly where you want a brand new one built, right from scratch. Here's how this molecular GPS system works. First, the scientists put a unique DNA sequence on a piece of clean, easy-to-read DNA. This is the address. Then, they send in a protein that's been engineered to act like a mailman. It's designed to find and bind only to that specific address. And here's the magic trick. They attach a special delivery package to that mailman protein. This package contains a key factor that tells the cell, hey, build a centromere here. The mailman finds the address, drops off the package, and the cell is basically tricked into building a perfect, fully functional centromere right where the scientists wanted it. And this just completely changes the game. We've gone from borrowing a mysterious engine from a junkyard to basically 3D printing a brand new, custom-designed one exactly where we need it to be. This really brings the whole story full circle. On the one hand, you have that fuzzy first-generation chromosome. It worked, but it was an unreadable black box. And now we have the blueprint for a sleek, completely defined, and fully engineerable chromosome. We finally made the leap from the black box to the actual blueprint. So why does this matter so much? Because now, for the first time, we have a chromosome where we know every single letter of its code. It's easy to build because you don't have to deal with that nightmare of repetitive DNA. And the big one, it is completely engineerable. It's a true platform where we can add huge pieces of genetic information and study them. This basically removes the single biggest obstacle that was standing in the way of creating fully synthetic human genomes. Which leaves us with a pretty profound final thought. For the first time, we have the tools not just to read our genetic code, or even to edit it, but to write entirely new chapters from scratch. So the question is no longer if we can, but what on earth we should write next.